Good afternoon and welcome to Genetics 311 Lightning Round video. We are going to explore the way that one single point mutation or a change in the reading frame that changes how a codon is read can change the entire sequence of mRNA interpreted and thereby the protein produced. Let's get started. First off, we are going to look at the DNA sequence. A protein formed from this DNA sequence in vivo will contain 5 amino acids if done correctly. In order to determine how the DNA gets transcribed into RNA, we must determine which strand is which, coding or template. We must flip the bottom strand to the 5' to 3' direction to get it oriented correctly. Upon doing so, we can see that both strands contain the key start codon that codes for methionine, denoted as ATG in DNA or AUG in RNA. The coding strand will have 5 codons prior to its stop codon, which would read as TAA or UAA. The 5 corresponding amino acids are methionine, valine, arginine, phenylalanine, lysine, and the stop codon, which in this case is TAA or UAA. Here are the different types of point mutations, transitions, and transversions, as well as the consequences of what these mutations can have on the sequences that they change. We will look more into the consequences and different specific types of mutations from the change of either one transition, which is a purine to a purine or a pyrimidine to a pyrimidine, such as A to G and C to T, or one transversion, which is a purine to a pyrimidine, or vice versa, such as A to T and C to G. A silent mutation is changing one amino acid to the same amino acid. In other words, there's no change in sequence. So from our original DNA sequence, changing the valine's 3 letters GTC to GUU, so just the C to the U, results in a transition. And this is a silent mutation because the GTC and GUU both still code for valine. A nonsense mutation is a stop codon made instead of an amino acid, which ends the sequence early. So the original DNA sequence for lysine says AAG, but the change of the first A to a U would result in the codon reading UAG, which is a stop codon. And additionally, A to U is a transversion. A missense mutation is when a different amino acid is made as opposed to the original. So a different sequence is made of the same length. The original DNA sequence would go from TTC to, when mutated, the mRNA being UUG. So phenylalanine would change to leucine due to the UUC being UUG. So a C to a G is also a transversion. A readthrough mutation is an amino acid instead of a stop codon, which elongates the sequence in strand length. So the original would say TAA, which would also be UAA for the last stop codon. But the mutated mRNA sequence, when the last A changes to a C, would read TAC or UAC after it had been transcribed into mRNA, which would be a transversion, but would also change it to be reading or TYR instead of a stop codon. So the mRNA sequence would continue on and add an additional sequence of multiple amino acids. Thank you so much.